Hi everyone, welcome back to this channel. So this video is about the third week of development. So basically in the previous videos we talked about the first week of development, the second week of development. So in the last video on second week of development, in that I have mentioned like in the second week like every two things or two layers are formed. So from the inner cell mass we had the epiblast and the hypoblast layer. Outer cell mass we had the cytotrophoblast and syncytial trophoblast layer. So in this video I'll talk about what are the things happening in the third week of embryo development. So let's get started. So in the third week two process takes place. So that is gastrulation and neurulation. It occurs in the third week of development. So gastrulation is the formation of the germ layer of the body, that is the endoderm, mesoderm and the ectoderm layers, and neurulation is the formation of neural top. So with this video I'll cover the gastrulation part, which is the formation of germ layer. So in gastrulation, first of all let's see the diagram. So here this red colored are epiblast cells, which are columnar cells, and these are hypoblast cells, which are cuboidal cells. So epiblast cells columnar, hypoblast cells cuboidal. So in the hypoblast cells, some cells like we can see in this orange colored, these cells are hypoblast cells, but from the cuboidal hypoblast cells they are becoming columnar hypoblast cells. So those cells are called procaudal plate. So procaudal plate is columnar hypoblast cells. So these procaudal plate, which are columnar hypoblast cells, it forms buccopharyngeal membrane, and this buccopharyngeal membrane will later on ruptures and form the oral cavity. So before the formation of procaudal plate, we just had epiblast and hypoblast, and we didn't know which is the tail end and which is the head end. So after the formation of procaudal plate, which is becoming the buccopharyngeal membrane and rupturing to form the oral cavity, then we know that this is like procaudal plate is forming the head end or the oral cavity. And this was happening in the hypoblast area. And now if we look into the epiblast cells, columnar cells, we can see this proliferation. So this proliferation of epiblast cells are called primitive streak. So primitive streak is a proliferation of epiblast. So the primitive streak is an indication that this proliferation of epiblast is an indication that the beginning of gastrulation has started. And also this will be the tail end, because procaudal plate was the oral cavity or the head end. So the primitive streak is formed in the third week, which is end of second week or the beginning of third week. So in some cases we will have this persistent primitive streak at the tail end, and it can lead to a teratoma, which is called Sacrococcygeal Teratoma. And this sacrococcygeal teratoma can be of two reasons. It can either because of the persistence of this primitive streak at the tail end, or also due to the abnormal migration of the primordial germ cell, which I have mentioned in. So that was it about sacrococcygeal teratoma and the two reasons why it can occur. So in this diagram we can see the procaudal plate, which further on forms the oral cavity and the head end. And here this is primitive streak, which was a proliferation from the epiblast. So these are this procaudal plate and primitive streak formation takes place. So again the procaudal plate formation, primitive streak on the day 15, which is like third week of development. So now let's see the gastrulation process, which is a formation of germ layer, and the first germ layer to develop it is the endoderm, and this endoderm is derived from the epiblast layer, and this endoderm will then replace a hypoblast. So after the endoderm formation, there will be no hypoblast seen hereafter, because all the hypoblast will be replaced by the endoderm layer, which is the first germ layer to get developed, and it is derived from the epiblast. Then after that, then we have the formation of second germ layer, which is a mesoderm layer, and this mesoderm layer is derived from the primitive streak, which was a proliferation of epiblast cell. So we can say that even the mesoderm is derived from epiblast, because primitive streak is derived from epiblast. And the last germ layer to form is the ectoderm layer. So basically, from the epiblast, first the endoderm layer will form, it will replace a hypoblast, and from the primitive streak of the epiblast, mesoderm will form. So afterwards, ectoderm will form, because the remaining of the epiblast which is left after the formation of endoderm and mesoderm, then the the ectoderm layer will form from the remaining of epiblast. So we can see that all the germ layers like endoderm, mesoderm and the ectoderm, all are germ layers are derived from the epiblast. So in like old editions, it was said like it was derived from yolk sac, but now in the latest 3 editions, it's been said like all the germ layers of the body is derived from epiblast. So this will be the answer. So in the diagram, we can see all the 3 germ layers. So this is endoderm, which is the first layer to form, and it is derived from the epiblast cells. Then afterwards, then we have the intra-embryonic mesoderm formation from the primitive streak of the epiblast, and all the remaining part of epiblast after the formation of endoderm and intra-embryonic mesoderm then forms the outer layer or the ectoderm. Now I'll talk about midline mesoderm. So this was a procaudal plate or the buccopharyngeal membrane, and this was a primitive streak which was the tail end, and the procaudal plate the head end. So in the midline, there is a notochord formation, which is a midline mesoderm. So notochord is derived from the midline mesoderm. So in the middle, that is this thing, it is notochord, which is also called midline mesoderm, and this notochord is derived from the primitive streak or the proliferation from the epiblast. So this notochord is derived from the primitive streak. And what is the function of notochord? This notochord structure will then stimulate the formation of neural tube, and this notochord in adult, it will not remain, it will disappear, and it will form some remnants. So there are basically 2 structures which are derived from the notochord, which is nucleus pulposus of the intervertebral disc, and also in some books it is also mentioned that from the notochord or the remnant of notochord, it can form the apical ligament of Dens as well. So basically, notochord will get disappeared in the later stages, and it will form the nucleus pulposus and the apical ligament of Dens. But in some situations, in some pathology, the notochord will not disappear, and it will persist in the adult life. And if notochord persists, then that condition is called chordoma. So chordoma is a congenital tumor where there is a persistence of notochord, and if to chordoma any word is added, like if it is nasopharyngeal chordoma or whatever chordoma, when the word chordoma comes, it means it is to be understand that it is because of the persistence of notochord. So it is a congenital tumor, and in chordoma we can see a special type of cells which is called Faciliferous cells. So in the diagram is for the Faciliferous cells. So these cells are having large vacuolated cytoplasm. The cytoplasm is with large vacuoles and nucleus is shifted to the periphery. So these are Faciliferous cells which are seen in Chordoma. So I said like there was endoderm formation firstly, then there was mesoderm formation which was intraembryonic mesoderm, then there was ectoderm formation. So there are some subdivisions in the intra-embryonic mesoderm and I'll talk about that. So first of all, this is a buccopharyngeal membrane, the procaudal plate and the primitive streak on the tail end, and in the midline mesoderm, which is the notochord, as mentioned, I have just mentioned before. So now we'll look into the mesoderm formation. So this intra-embryonic mesoderm, there are subdivisions. First of all, there is this one which is midline, which is like nearby to the midline mesoderm or the notochord. So that is called paraxial mesoderm. Okay, and then we have this one which is the intermediate mesoderm, and then the rest of the mesoderm is called lateral mesoderm. So in lateral mesoderm also there is a cavity formation which is a blue color, and this, because of the cavity formation in the lateral mesoderm, it is dividing this lateral mesoderm into 3A and 3B. So 3A corresponds to splachnopleuric layer of lateral mesoderm or the visceral layer of lateral mesoderm, and 3B, that is this one, is a somatic layer or parietal layer of lateral mesoderm. So these are the subdivisions of intraembryonic mesoderm. So the paraxial mesoderm, which is this one which is lateral to notochord, and then the intermediate mesoderm, then the lateral mesoderm, which having the somatic layer or the parietal layer of lateral mesoderm and the splachnopleuric layer of lateral mesoderm. So these are the subdivisions of intraembryonic mesoderm. Now I'll talk about from each subdivision of intraembryonic mesoderm what all structures are formed. So from the intermediate mesoderm, which is this one, the urogenital system is formed, and from this splachnopleuric layer of lateral mesoderm we have the smooth and cardiac muscle formation, also the splachnopleuric layer of body cavity, which is a peritoneal, pleural, pericardial cavities. And from the somatic layer of lateral mesoderm, we have the appendicular skeletal formation. Appendicular skeleton means the upper limb and the lower limb bones, as well as the somatic layer of all body cavities. So in this diagram, we can see the different layers, the endoderm, the ectoderm, and the mesoderm. So in the mesoderm, we can see the subdivisions, the notochord in the center, and near to notochord we have the paraxial mesoderm, and this paraxial mesoderm is then forming somite. I'll talk about that. And after the paraxial mesoderm, we have the intermediate mesoderm, from which the urogenital sinus system is formed. Then, so we can see that when we also have the lateral mesoderm forming the splachnopleuric mesoderm and the somatic mesoderm. So from the splachnopleuric lateral mesoderm, we have the smooth and cardiac muscle formation and the splachnopleuric layer of body cavity, and from the somatic layer, the upper limbs and the lower limb bones is formed, as well as the somatic layer of all body cavities. So from the paraxial mesoderm, somites are formed. So the first pair of somites which are formed from the paraxial mesoderm is happening in the occipital region, and it is formed in the 20th day. And from afterwards, from the 20th day, after each day, 3 pairs is added. For example, in the 21st day, from the first pair, 3 more pairs are getting added, so 4 pairs are formed. On the 22nd day, 4 plus 3, that is 7 pairs are formed. So on each day, like 3 pairs are getting added. So the 23rd day, it will be 10 pairs. So like on the 35th day, like in 23rd day, it was. So if we see on 35th day how many pairs are formed, like if you minus 35 minus 20, like after 20th day, 35 is like 15 days, so 15 into 3, like each day 3 pairs are formed. So approximately 42 to 44 pairs of somites are formed on the 35th day, which is the 5th week. And the order of appearance of somites is like the first somite to appear will be on the occipital region, then the cervical region, then the thoracic, lumbar, sacral, coccygeal. So we can see the pattern, like the appearance of somite is from the craniocaudal sequence. So after 5th week, what happens is that some somites from the occipital as well as from the coccygeal region will disappear. So the approximate number of total pairs of somite after the 5th week, because the occipital and coccygeal pairs get disappeared, will be 37 pairs. So let's see the diagram. We can see this is paraxial mesoderm. So in paraxial mesoderm, we have the somite formation. Okay, and this somite also, there are division inside the somite. So it is a sclerotome and this is a derma-myotome. So somite is getting divided into sclerotome and derma-myotome. And the sclerotome will form the axial skeleton, which is the vertebra, and we can see that the sclerotome will go around. Okay, so it is going around the notochord or the neural tube, and it will fuse with the opposite side. It is going around the notochord, which is this one, neural tube, and it is fusing with the opposite side. So because of this going around and fusing in the opposite side, the sclerotome part of the somite will have different parts. So this is a ventral end of the sclerotome, then we have the dorsal part of sclerotome as well as the lateral part of sclerotome. So sclerotome in total is forming the axial skeleton, which is a vertebra. So the ventral part will correspond to the body of vertebra, and the dorsal part will correspond to the spine of vertebra, and the lateral part will form the vertebral arch. So if the ventral part of the sclerotome fails to fuse, then we will have a condition called hemivertebra. Whereas if the failure of fusion is in the dorsal part of the sclerotome, then we have the Spina Bifida anomalies. Okay, so we have like Spina Bifida anomalies. It is like of 3 kinds. So there is Spina Bifida occulta, then there is meningocele and myelocele. So in Spina Bifida occulta, it is the failure of fusion of the dorsal sclerotome, and a tuft of hair can be present over the defect. Whereas in case of meningocele, there is dorsal sclerotome failure to fuse as well as the meninges is protruding over the defect. In myelocele, along with the meninges, there will be spinal cord also protruding from the defect because of the failure of fusion of this dorsal sclerotome. So in the dorsal side, failure of fusion can lead into Spina Bifida anomalies, whereas if the ventral side of the sclerotome is failing to fuse, then we have hemivertebra. So the picture A, this is corresponding to Spina Bifida occulta, which is a failure of fusion of the dorsal sclerotome. The defect is here, and there can be tuft of hair present over the defect. Then this is meningocele, where there is protrusion of the meninges also, meningocele, and myelommeningocele or meningomyelocele, then there is spinal cord also protruding along with the meninges through the defect. So as I said, from the paraxial mesoderms there is somite formation, and somite is also divided into sclerotome and the derma-myotome. Okay, so now previously we saw the sclerotome layer, and it is corresponding to vertebra formation, the dorsal, ventral and the lateral part. And now let's look into this dermomyotome layer. So this dermomyotome is also divided into two. So there is dermatome and myotome formation. So myotome, as the name suggests, the skeletal muscles of the body will be formed from this myotome layer of paraxial mesoderm, whereas dermatome, it is forming the dermal layer of the skin. So the endodermis of the skin is formed by the surface ectoderm, whereas the dermis layer of the skin is formed by the dermatome. Okay. So till now what I mentioned, like from each subdivisions and what all structures are formed, that all we have to know and we have to memorize it, because questions will come. Like for example, the biceps of muscle, so it will be asked like from where it is formed. So bicep will be formed from the myotome layer of the somite, and this myotome is of the somite, and somite is from the paraxial mesoderm, whereas the humerus bone, it will be formed from the somatic layer of the lateral mesoderm. So such questions comes, so we have to know the answer. So while mentioning the splagnopluric layer of lateral mesoderm, I said that it corresponds to the cardiac and the smooth muscle formation. But there are some exception, like there are some kind of smooth muscles which are formed not from this splagnopluric layer of mesoderm but from other structures. So I'll talk about that. So the smooth muscle over the dorsal aorta, it was previously said it was formed from the paraxial mesoderm, but now, like other smooth muscles, it is also formed from the splagnopluric layer of lateral mesoderm. But when question come like the smooth muscle over the pharyngeal arch arteries, then it will be from the neural crest cells. And smooth muscle in the iris, which is frequently asked, it is derived from the neuroectoderm layer, and the smooth muscle over sweat and mammary glands are derived from surface ectoderm. So these are something to be noted. So that was about gastrulation. Now in the third week of development, two more important process takes place other than gastrulation and neurulation. Neurulation I'll talk in the future video. So the other two important processes, angiogenesis and hematopoiesis, which is taking place in the third week. So what is angiogenesis and what is hematopoiesis? So angiogenesis is the synthesis of blood vessels. So and from which germ layer the blood vessels are formed? It is derived from the mesoderm. So the mesoderm differentiate into blood vessels, and thereby blood vessels are formed. So I have mentioned in previous video also, like there is primary villus formation and secondary villus formation. So primary villus is formed when the cytotrophoblast invades into the sincere trophoblast layer. These are basically the two layers of trophoblast. And the secondary villus is formed when the extraembryonic mesoderm, and which will be the extraembryonic mesoderm? The somatic extraembryonic mesoderm. When that invades into this primary villus, so we have the mesoderm core, then we get the secondary villus. So then how we get the tertiary villus? So now what I am saying is that the mesoderm will get differentiate into blood vessel. So some of this somatic extraembryonic mesoderm, which is in the mesoderm core, will differentiate and forms this blood vessels. We can see the red color. So once the blood vessels are formed, then we have the tertiary villus formation. So the tertiary villi will contains blood vessels, the somatic layer of extraembryonic mesoderm, the cytotrophoblast and syncytiotrophoblast. So the secondary villi were containing all these three things, plus the blood vessels will form the tertiary villi components. So I said mesoderm differentiate into blood vessels. So which mesoderm will differentiate, because we have many subdivisions of mesoderm. So first mesoderm to differentiate into blood vessel is the splachnopleuric layer of the extraembryonic mesoderm, which is the first mesoderm which differentiate first. Then comes the somatic layer of extraembryonic mesoderm. Then afterwards comes the intraembryonic mesoderm. And also the other process which is taking place in the third week is hematopoiesis. Angiogenesis was a formation of blood vessels, whereas hematopoiesis is the synthesis of blood cells. So on the third week, which is forming this blood cells, or what is causing the hematopoiesis, it will be the extraembryonic mesoderm, the splachnopleuric layer. And after the fifth week, the hematopoiesis function is taken up by these organs, which are LTSP is the mnemonic. So it is liver, thymus, spleen and bone marrow. So and this liver will take up the function of hematopoiesis only up to 7 months. So that is the end of this video. Hope you could understand it. Thanks for watching the video. Please support the channel by sharing and subscribing to the video. Thank you.